the very first thing we did was we got our Bacillus subtilis bacteria, which is gram-positive, and we extracted the chromosomal, that genomic DNA, okay? And I wanted to highlight what some of the major steps of the procedure were and its importance. So the first thing we did when we got our culture was to spin it down. So this just basically concentrates our bacteria. And then the first thing we did after that was lysing the cells so we can have access to our DNA, okay? And we did this with an enzymatic lysis buffer. What this lysis, buffer is, these enzymes are, their entire initiative is to destroy, okay? So destroy everything that surrounds the outside of the cell. And this needs to be particularly destructive, especially because it's gram-positive, okay? And so my question to you guys is, why does it matter if it's gram-positive? What aspect of gram-positive bacteria make a DNA extraction particularly difficult, let's say, compared to like a colony DNA, or if we were to simply use bacteria that have not been DNA extracted, like when we did colony PCR, we didn't have to do this rough lysis of the bacteria. So what aspect of this, of gram-positive bacteria, make it rougher to get that DNA out? Yes, so it's going to be that cell wall, that thick cell wall, the peptidoglycan layer. Okay. So I want you to keep in mind that this is genomic DNA extraction. Okay, and there's two types of DNA extraction. This is just genomic chromosomal DNA extraction. It's different from plasmid DNA extraction, which we'll be looking at later. So after we lyse those cells, we washed them, we cleared them. All the while, the DNA is stuck to that column, right? It's stuck to that column. And only at the very last moment, when we've already washed all the DNA, that's when we elute it, okay? And then we'll have our hopefully pure DNA. I have a question. Is that the reason we have to use enzymatic lysis buffer? Yes, I mean, you could also use it for gram-negative as well. It should be fine. But I do want to highlight that this lysing step uses the enzymatic lysis buffer, and it's going to be different than the plasmid extraction, which we'll talk about later. Okay. So what did we do once we got that DNA extraction? The first thing we want to do is we want to look at the quality of that DNA we just extracted. We can measure it. We can quantify what the concentration is, what the purity is, looking at the 260/280 ratio, which is just looking at the absorbance through the spectrophotometer. That'll tell us what ratio there is between those two wavelengths. And so we know if there's any protein contamination and things like that. And those wavelengths are particularly absorbent because of those aromatic rings in the DNA compared to the aromatic rings of proteins at 280. Okay, and so again, quick question: what's that number we're aiming for in purity? That 260/280 ratio. What are we aiming for there? Do you guys remember? Yeah. Okay, good. That 1.8. Okay, so once we had our genomic DNA extraction, we then went on to PCR amplify our specific gene of interest, which I've highlighted over here in red, the alpha amylase gene. Our goal is to take this alpha amylase gene from Bacillus subtilis into E. coli, so we can extract that protein at the end. We're PCR amplifying this gene now. And so what this mixture entails is we have our Taq polymerase. Okay, that's going to be doing all the work for us, and reading our primers to extend the DNA, or reading the DNA, using those primers. So what does that Taq polymerase require? It requires a specific buffer as well as cofactors like magnesium to work properly. Additionally, Taq polymerase, if you remember, works under extremely high temperatures. And this is really important because we're going to need to use really high temperatures for that denaturation. And so we don't want to ruin our protein in the process, our polymerase in the process. Other things that we need to add to our PCR mix is our DNA, of course, that has hopefully our insert in there, that alpha amylase gene. And then we have a forward and reverse primer. Okay, and we'll talk about primers in a second. So we have the first step in PCR amplification. We have all of our chromosomal DNA in there. We need to fully denature all of our DNA, okay? That will allow those primers to go in, okay, and anneal in the annealing step, okay? So those primers, like over here, will anneal to, the top and bottom strand, okay? This will be where that Taq polymerase binds and extends the fragments, okay? And so this process will go on and on until you have billions of copies of your specific DNA fragment. There are really good videos that you guys can watch. They're really short videos that kind of animate the PCR product or the PCR process. So you can see why the primers allow only this fragment to be elongated and only this fragment to be amplified. Let me know if anyone had a question. Sorry. You guys good so far? Okay. So we've annealed, we've added these primers, that Taq polymerase is extending. We're going to repeat this process 30 times usually, so that denaturation, so detaching of our two top and bottom strands, primer annealing, and then extending that strand. Okay. So important things about primers. They have to be designed properly. And there's a lot of websites that can design them for you, so don't stress out too much about that. But you need to be able to troubleshoot sometimes if you're not getting good bands, okay? And that's usually due to primers, and we'll talk about that. A good melting temperature is around 52 to 58 degrees. And I want you guys to think about what would happen if the annealing temperature, okay, the annealing temperature, the temperature at which the primers bind back to the DNA, okay, what would happen if that was too low or too high? Okay, so first, if the annealing temperature that we set our thermocycler to was too low, okay, what would we see on a gel? How many bands? Would there be a band at all?
Is this the same thing as the melting temperature?
Yes. Yeah. Kind of, yeah. It's a little bit confusing, but it should be called the annealing. Because melting indicates we want them to melt, but annealing here, we're trying to get them to come back.
So when it's, if I remember, when it's too low, usually he said that the primers would not attach at all, because they become they don't want to flow in a sense, they need heat in order to find the location they need to attach to. So whenever it's too low, they don't have the right like environment for them to actually get to work. So there will be no bands.
I'm not sure about that. It sounds more like he was talking about Taq polymerase. Taq polymerase needs to be at a really high temperature to work. The primers, let's imagine, yes, Ariana. Yes, there were a lot of bands. We're going to talk about that. So let's imagine our temperature, our annealing temperature was too low. Okay. And our melting temperature was like 52, and our annealing temperature was way below that. Okay. This actually allows, well, I don't want to answer my question. I want you guys to answer why. Ariana's right. Why? Okay. So in the chat, she said, at lower temperatures, there's a lot of bands in your PCR. After you do PCR, when you load that onto a gel, you end up with a lot of bands. The only reason you could end up with a lot of bands is what? What do you guys think?
I don't really know, but would there be, oh, never mind. Yeah.
Less specificity. Okay. So exactly. There aren't there several primers that make their way towards the template strand or something like that? Yeah, I think Brandon, you're alluding to the same thing, which is that basically the primers bind non-specifically because they're able to at such a low temperature. I think it's because of the bonds allow them to be more stable at a lower temperature, and so it doesn't need to bind perfectly. And so that's why there's a lot more non-specific binding. Okay. So if we had the right annealing temperature, then we would only get one band, right? Because we're only trying to amplify one gene. It should have one size, one band. What if our annealing temperature was way too high? What would happen then? So we want them to anneal, and we put the annealing temperature higher than the melting temperature, hotter than the melting temperature. So Ariana is completely right. You're not going to get any bands at all because none of them are going to get the chance to bind or hybridize back with the DNA. The temperature's too hot. It's past their melting temperature. Okay. Let me know if this doesn't make sense.
I have a question about the whole temperature. When you mean the primers don't, like when it's too hot, they can't bind, and when it's too cold, it's too easy for them to bind to different spots.
Yeah.
But how come it becomes easier? Is it because the parts of the DNA, like why does it become so easy when it's a low temperature? Like why? Like what about the DNA and the primers interacting when it's low temperatures? Like I don't understand that part. Is it because you said something about bonds, but what bonds are we talking about?
My guess is that the hydrogen bonds become more stable, and they either don't need all of the bases hybridizing together or to be stable enough, or there's like non-specific binding, hydrogen bonding. I don't think that's as likely. I think the primers just like don't completely, not every single hydrogen bond is there, but it's stable enough so it doesn't fall off. That's my guess, but it's not, it's
Okay.
not like he won't expect you to know why or anything like that.
Okay.
Mhm.
So just know low temperatures, more bands,
Mhm.
more temperature, less bands. Gotcha.
Yeah. Okay. Any other questions so far? Okay. I apologize if I'm going a little bit fast. It's just because we started a little bit late, and I don't want to cut into Dr. Gonzalez's discussion thing later. Okay, so once we've done PCR, okay, now we're ready to visualize that on a gel, okay, which is what we did. So we visualized that, we got one band. What is that gel made out of again? We have that 1% agarose, we have that TAE buffer, we have our gel red fluorescence dye that will intercalate, you know, and stack along the DNA rings. And my question to you guys is, what if the agarose percent was different? What would happen to the band? So let's say we had a higher percentage of agarose. Let's see, how would this gel look different? Would we have more or less resolution? So let's say that this band was very small, okay? Yeah, absolutely. So if the band was small, then increasing the concentration of agarose, making it thicker, will allow us to have better resolution of smaller bands, such as our PCR product, okay? If we had a really long piece of DNA, okay, let's say our piece of DNA was super big, okay, like many kilobases. If we increase the agarose percent, if we made it thicker, you know, more concentrated of agarose, it would be harder for that long piece to get through, and we'd get less resolution. So for larger bands, we want to dilute that agarose, we want to decrease that concentration, decrease that percentage. But for small bands, we want to increase that percentage, so it doesn't, you know, run off quickly, and we can see it clear. Okay. All good. Okay. So now that alpha amylase gene is ready for cloning, but before we attach it to the plasmid, we got to figure out how we got the plasmid. So we started with our plasmid extraction. So we originally had a culture of E. coli, okay, that had our plasmid inside. So we needed to get that plasmid out, which is right here. The first thing we did, spin the culture down, concentrate those cells, and to remove the LB that it was in. And then we lyse the cells, okay? And this process is much more different than, is very different to this enzymatic lysis process, okay? So here we have that alkaline lysis, okay? This is much gentler. And has a mechanism, by the way, because plasmids are they're smaller, and we need to have a more gentle, I don't know, lysis, I guess. But what's more important is to separate the chromosomal and plasmid DNA. What this protocol also allows is this reanneal process where this alkaline lysis will denature. Even our plasmids, our DNA, all of it actually will denature it, but not break it. It will just denature it. And the next step right after lysis is to, I don't know, it's to add a buffer that will allow the DNA to reanneal. The plasmids will reanneal real quickly. The chromosome will take a very long time to reanneal. And so that allows you to just get that, that plasmid instead of all the chromosome. I had a question, is the E. coli DH5 alpha where we extracted the plasmid? I don't think so. I think it was just E. coli. I don't know specifically what strain. So the DH5 alpha comes in later for transformation. Okay. And then after that, we have the BL21, which we're not getting to this midterm, in this midterm. But yeah. So that's basically the process. For plasmid extraction, we use the alkaline lysis, which is gentler. That's pretty much what you need to know. And then it allows for the chromosome to be discarded while the plasmids are able to reanneal quicker and then be extracted. So we go through those wash steps again, and then elute it at the end. So let's talk about what this plasmid is, what's going on over here. So this plasmid has that SACB gene that we know, with its own promoter and everything. That SACB gene, as you guys know, is lethal to the cell. Okay, so we have that suicide, this is a suicide vector. And it also contains ampicillin resistance. We have our ORI, so it can replicate. We have our LAC-I, which is for later. That's for regulating the expression of this protein. And the multiple cloning site. Okay, so this SACB is, its purpose is to let us select clones. Okay, let us select recombinant clones. Okay, let us select E. coli that have not just taken up the plasmid, but have also been inserted the gene. Okay, which is going to be right over here. And then ampicillin resistance allows us to select for any E. coli, any bacteria that has taken up the plasmid at all. Okay, so that multiple cloning site has some important characteristics there. We have our T7 promoter. We have our his tags, that's for later. That his tag allows us to purify the protein. We have our LAC operator, which is that allows us to regulate that expression again. We have those restriction sites, which we'll talk about in a second. We have thrombin. Thrombin just allows us to cut off the his tag. Okay, so don't forget, thrombin is an important part of our multiple cloning site there. Allows us to cut off our his tag. And then not just do we have the T7 promoter, we also have the T7 terminator there. Okay. If we were to run this on a gel, what would we see? Okay, we end up with 3 bands. And do you guys remember why we get 3 bands when we're only loading one piece of DNA on there?
Different forms of plasmid, right?
Mm-hmm. Exactly. So there is the supercoiled, the linear, and the circular. I don't think the order is so important for this class, and I always get them confused because it's not like it's not intuitive which order is which. The supercoiled is definitely the last one, and then I believe the top one is circular. Let me double check.
No, I think the linear is the middle one.
Thank you guys. You guys know. Good. Okay, so we end up with those 3 forms.
I have a quick question about the promoter and the the multiple cloning sites.
Mhm.
So he talked about the his tag on the review on Tuesday, and he talked about how the his tag is just like the like the purification part, like tag. I think that's what he tried to explain, and how the sticky, it has like sticky parts where it connects to the to like the peptides, like the thrombin and the, I believe so. And then why was the reason that you caught that? Is it because of the when you have to do a recombination, or?
Yeah, so the his tag, like you said, when you go to purify your protein, that his tag will get stuck in like a column. And so that means this column will be full of only your protein of interest, because this his tag, this histidine tag, will bind to nickel on the column, okay, to purify, okay? And so that'll allow you to only pick one, like to purify only one protein, your protein of interest. And then when you finally get your protein of interest all purified, well, the his tag was not naturally there, right? The his tag's not naturally a part of our protein, and we don't know what kind of effects it'll have when we test this protein. And so we just want to cut it off. That's what the thrombin site is for. Does that make sense?
And that's after we add the amylase, or that's either before or after?
That'll be after. So this thrombin site is sitting there. These are all like just sitting there in preparation for our gene of interest to be inserted. So I don't think I have a good picture. Yeah, I don't. But basically our gene of interest will sit over here, and it'll actually, I wish I could show you a better picture. But basically all these aspects here are waiting and sitting there and ready for the gene to be inserted. And so yeah, they're already there and then can be used after the gene's inserted too. Does that kind of make sense?
Like it's kind of like the hot spot where genes know where to relocate again and where to like cut, like where whenever we do the transformation, not transformation, the extract of like specific genes, because we're gonna eventually remove SAC-B, right? And those spots are like there to kind of guide or like what was like that we were using in order to extract it. I don't remember the restriction sites. And once we have to insert it back, there's like they're like the, well, how I think about it, it's like the ending parts that we need so it can recognize it, right? From the plasmid when it's open, right? Those are the sites where it can recognize it. And then those are the sites the same way that so they know where to extract. That's how I think of it, right? Okay. Then
yeah. So this ND1, this BSER1, you can think of those as sites to cut. Okay, so we're going to be cutting here and cutting here. I have it here in my restriction digest. So we'll be cutting here and cutting here. Okay. Our goal is to then insert our alpha amylase gene right into there, you know, with those sticky ends. And then our his tag thrombin site, T7 promoter for this gene, T7 terminator for this gene will all be there, you know, and ready to use. So
I have a quick question about, you said sticky ends, right? I was absent for that kind of lecture, but my lab partner was telling me how the sticky ends are usually something that you see on the restriction enzymes, but not when you're doing recombination, homologous recombination, right? Or is that something that was not specified?
Okay, hold on. So are you familiar with what sticky ends are?
Yeah, when it gets snipped out, so those are the areas where kind of like the primers will work and start like inserting the sticky ends where you reinsert the gene that you want, which is the alpha amylase or whichever you want to add, and those are like the sites where it's going to start complementing it
Mhm.
and then complete the insertion of that genome there. That's what I got like from reading the PowerPoints.
That's true, but also specifically sticky ends are simply just ends where, so notice this line I drew here is where the restriction enzyme will cut. Okay, so if you notice, there's going to be flanking ends. Do you know what I mean?
Like overhanging.
Exactly. So those sticky ends are simply just overhangs and flanking ends. So that's really what it is. And that allows you to insert the gene, which should also have the sticky ends on each side. I should have
Yeah, the
known.
complementary ones, right? I think.
Exactly. So let's say there's a stick. Oh my God. Hold on.
Because also I said that they talked about overhangs for primers as well. Like the overhangs are usually after the primers, reverse and forward.
Yeah, so during PCR, you do get overhangs for I think like after the first round of PCR amplification. And that's just because let me show you a picture, hold on. But yes, you're right, you do get PCR overhangs.
Yeah, like my only concern is with that is like, what is the difference between those type of overhangs with the overhangs after the removal of that section, like the SAC-P?
They should, like, whether they're sticky ends or not.
Yeah, like are the ones that are overhanging on the PCRs the same as when you see in restriction enzymes? But because the way how my lab partner kind of explained to me is whenever you're trying to recombinate the plasmid with the gene, the alpha amylase, you will have some kind of like overhangs, but they're not sticky ends overhangs. They're more for like recombination. And they have primers, like the primers are still there, but they have like the sticky ends will be kind of gone. That's what he said. So, but now you're saying that the amylase will have like those sticky ends, and they will use those sticky ends to kind of implement themselves into the plasmids.
Yeah.
So I'm not sure if like, so sticky ends can be seen whatever there's a split on the genome.
So yeah, sticky ends simply just refers to like whenever there's an overhang, like a single strand overhang of DNA, which is usually caused by like restriction enzymes cutting it. We did actually do a restriction digest of the alpha amylase. Dr. Gonzalez said we didn't need to do it, but he just did it in case. But honestly, I think we needed to do it. So that's why I didn't include it in my chart here, because I wasn't sure if like it was necessary, but we did do it. So we also digested our alpha amylase so that theoretically it created sticky ends at the ends here too, so that it could be perfectly inserted in here and attach and hybridize. So yeah, I'm not sure what Sebastian was talking about specifically. But I mean, in PCR, there are overhangs that are not necessarily sticky ends. That is true.
Yeah, because I think the confusing part was whenever we talked about ligase, because it's ligase independent cloning. He said that we didn't do that one. Like it did take part, but we didn't perform it. And he said that what happened was more of a homologous recombination instead of a ligase cloning, like independent cloning, which I was confused about. Because I was like, so we didn't do that, did we? Maybe the confusion is where he said that we didn't need to do that. Maybe he understood it wrong, but that's what he explained to me.
Because we did do ligase independent cloning. Actually, we use Gibson.
Mhm.
I don't know if it was Gibson cloning or not, actually. Just in case, I'm gonna remove that. But we use ligase independent cloning. And it was a specific enzyme, which you guys don't need to know, that was basically able to connect these. Oh, I know exactly what you're talking about now.
Okay.
There was recombination here. That's exactly what happened. So instead of ligase going ahead and like just like gluing the PCR insert, the alpha amylase, to the plasmid, there was like
Homologous recombination, I think.
that was able to attach these together.
And that involves the sticky ends as well?
Yes, I think.
Okay.
Yeah, I will double check this, and we'll talk to Dr. Gonzalez about it. But anyway,
Yes.
that is not a huge focus. That's why, like, I'm not gonna go too much in depth into that. But what is important is that there are sticky ends on the plasmid, and this allows for directional cloning. Okay, so if you were to choose restriction enzymes, that created blunt ends. So if there was a restriction enzyme that cut like that, you know, because every restriction enzyme is different. Some like to cut like this. Maybe another one will cut like this. Maybe another one will cut like this. Okay. Oh yes, Marina, Dr. Gonzalez will be having another discussion at 7 p.m. Okay, I'm gonna try to wrap this up as much as possible, so you guys can go ahead to that if you'd like. But we don't have too much after this, yeah. So if your restriction enzyme were to cut bluntly right in the middle, so there's no sticky ends, there is no telling in which direction will your insert go into here. Okay, it could flip backwards and forwards or whatever. So that's why it's important. Usually you should be using restriction enzymes with sticky ends. Okay, so I answered this question here for you guys. So why use two different restriction enzymes or restriction enzymes with sticky ends rather than blunt ends? Okay, that allows for directional cloning. Okay, you guys should also be comfortable with restriction enzyme digest and analyzing that on a gel. Okay, so here's another example. Let's say there's a restriction site A, B, and C. Okay, so if you were to visualize those cuts on a gel, what would it look like? Okay, so the first column, the first well, would just be one cut or no cuts. Okay, if you were to cut, let's say, A and B, you would get two fragments. Okay, you cut twice, so you'd get the fragment in between A and B and the fragment, you know, between the other fragment that's larger. So make sure you guys are calculating these sizes because it can get confusing. Okay, if you're doing this on a question, just make sure you count how many bases each fragment should be. Okay, that's really important. And then this third column would be as if all three sites were cut by their respective restriction enzyme. Okay, so sorry, I'm going to speed through here. Then we have the cloning, which we kind of talked about, that ligase independent cloning. And we'll clarify that more with Dr. Gonzalez right after. But I'm sorry, I meant to erase that. Okay. So, yeah, this is also just visualizing before we had inserted our insert. This is just after our digest, this gel here. So we have the vector plasmid, and then we have that sac B that we cut out. Okay. Are you going to be at the review with Dr. Gonzalez? I can sit by for sure. Okay. So the whole point of doing this restriction digest is, number one, to cut out our lethal sac B gene, and number two, to get our insert in there. Okay. So then transformation comes in, okay? And so transformation is simply getting our plasmid inside E. coli. What kind of E. coli? DH5 alpha, which is a cloning vector, okay? So bacteria that is suitable for cloning and for making high copies of our plasmid, okay? The technique that we use is called heat shock. Basically, this freaks E. coli out into uptaking whatever DNA is around them, so that hopefully maybe they can get, a gene that will help them survive this terrible stress. So they'll take up all these plasmids for us, and then we'll grow them, okay, on SOB or SOC. So that's that really, that's that recovery broth, okay, like the super optimal broth or the super optimal broth with catabolite repression, which is just like glucose for E. coli. Why did we do that? Okay, I want you guys right now to write in the chat why it's important that we recover these E. coli before we simply just plate them on a plate with agarose or with agar, sucrose, and ampicillin. Okay, why did we recover these cells for 1 hour before we plated them onto here? Okay, don't forget that. Okay, so we grew them, we recovered them for 1 hour, not because they're necessarily stressed out, because they were, and we do need to nurture them back to health. But the most important part is we need to let them express ampicillin. Otherwise, we will never get any colonies on our agar plate, okay? Because none of them had the chance to get that resistance gene expressed, okay? And then last step what we did is colony PCR, okay? So if you remember, originally we did PCR with extracted pure DNA, okay? Here, we're literally taking a dirty colony and we're just throwing it in our PCR, okay? And so do you guys remember the reason we're able to do that? I kind of mentioned it earlier, is because we have gram-negative bacteria this time. And so the membrane's way easier to penetrate. And so it works out. And so when we do colony PCR, we're going to be using the same primers we have been, same primers to simply amplify our alpha amylase gene, okay? So that's pretty much the process that we went through.